there's a little indicator that kind of shows the volume, the mic picking up my voice, and it doesn't look like it's hardly moving. I don't know what's going on. Okay, so tonight, chapter 14 is lipid metabolism, and then we're going to look at 15, which is protein metabolism, and then at the end, we'll see a summation of the whole shebang that we've been looking at the last few weeks. So, lipid digestion. So, about 98% of our dietary lipids are triglycerides. If you recall, these are also called triacylglycerols. These can even be abbreviated as tags. We'll use these terms all interchangeably. When we eat them, because they're water insoluble, very little happens in the mouth with the saliva. In the stomach, there's a churning process that happens that breaks them up into small droplets that are called chyme. So this is a physical process, it's not a chemical process. High fat foods stay in the stomach longer, making you feel longer. This is why some people, when they're doing high protein, low carb diets, they go for more fat than normal for that very reason. In the stomach, we do have gastric lipase enzymes that will hydrolyze about 10% of those triglycerides or tags. As it moves into the small intestine, however, I gotta add people to the meeting here. Guess I should keep that open too. Okay. So as it moves into the small intestine, the presence of this chyme triggers a release of bile that's in the gallbladder. And bile, we had talked about before, is an emulsifier. So this will act as a way of taking these free fatty acids and forming micelles around them. You remember we looked at this before. You've got these polar heads with these nonpolar interiors. These tails are nonpolar. And so fatty acids will fit in there and make these micelles, which can then pass through the intestinal wall. And our pancreas produces lipases as well that will continue the hydrolysis of these fatty acids. It rarely does a total and complete job, though. So here we have our triglyceride. Remember, it's a glycerol with 3 fatty acids hanging off of it. It will get hydrolyzed with the presence of a lipase, and then we will get some of the fatty acids coming off as free fatty acids now, but not all of them will necessarily be removed. And so this is not a glycerol, it's a monoacylglycerol, referring to this fatty acid that's still on there. Inside the intestinal walls, these fatty acids are reformed back into triglycerides, believe it or not. So it like breaks down in order to transport through the wall, and then it restructures as triglycerides. And these get formed as lipoproteins, which are called chylomicrons. So over here, I put a picture. You can see this huge jumbled collection of yellow structures. These are your tags inside of here. And you have these membrane lipids are the orange and green, and there's also proteins involved here, but these kind of form these little balls, if you will, of these triglycerides, and these are called chylomicrons. And these pass into the bloodstream through the lymphatic system. The lymphatic system is this green network that you see in the picture here of the body, and you see that there are these little lymphatic nodes that are here in the groin area and also the armpit area. You can see quite a few here around the intestines as well. And there's even some up in the neck. But these are this is how they get into the bloodstream, is through this lymphatic system. In the bloodstream, the triglycerides once again get hydrolyzed to free fatty acids. So this happens all over again. And if they're not immediately consumed, if these free fatty acids are not immediately consumed for energy, then they get stored in what is called adipose tissue. And an adipose cell is called a adipocyte. And this is a cell that is made up mostly of a globule, can't say the word right. Globule, thank you. Got her now. Globule of triglyceride. There's some cytosol inside this cell, and there is a nucleus, but most of this adipocyte cell is just carrying this triglyceride. These are some of the largest cells in our bodies. And like we said, most of the cytoplasm is replaced by this globule of triglyceride. Adipose tissues are mostly beneath the skin, and they're used to store energy, but also protect organs from heat loss and shock. So this is actually a good thing to have some of these. If you get too much, then you're fat. But if you need to have at least some of these, because they do help you store energy, and they also will help you against your organs being damaged or something of that nature. So here's the overall process. We ingest these dietary triglycerides into the mouth. Not much happens there. In the stomach, we have this churning action, which produces the chyme, these small fat droplets. We do get some hydrolyzation of the triglycerides with gastric lipases. These move into the small intestine, where the bile comes in and will act as an emulsifier so that these droplets can be brought into the bloodstream. And we do have more lipases that come from the pancreas, which will break them down and form these fatty acid micelles that we just looked at. These will move into the intestinal cells, where they get repackaged back again into triglycerides and form those chylomicrons, which can then pass through the intestinal wall into the bloodstream via the lymphatic system. So it moves those triglycerides into the bloodstream, where they get broken down again, and we end up with free fatty acids in our bloodstream that can now either be used immediately for energy or can be stored in those adipocyte cells. Any questions there at all? Okay, tag mobilization. When your body needs energy, there are hormones like adrenaline and glucagon will trigger the hydrolysis of these fatty acids in order to get energy. And trigger the hydrolysis of the triglycerides to get you free fatty acids, which can then be consumed and to form energy. There's a 3-step process. You have to activate the fatty acid, transport that fatty acid elsewhere in the body, and then it gets oxidized. We'll look at these 3 steps. In the activation, the fatty acid gets converted into a high energy acyl CoA. If you remember, we talked a lot about acetyl CoA. This is the product that ultimately feeds into the Krebs cycle. The term acyl just means that it's not just 2 carbons. Acetyl CoA is a 2-carbon CoA. In this case, we have a large R chain of undetermined length, and so the term acyl is used for that instead of acetyl. So there could be many, many carbons chained out here on this end, but it will react with a coenzyme A in order to form this acyl CoA. So this is activating that free fatty acid. And then it needs to be shunted into the mitochondrial matrix in order to be utilized for energy. And the mechanism for doing this is it uses a chemical compound called carnitine. It will react with this acyl CoA to put that acyl group from the acyl CoA here onto the carnitine and form acyl carnitine. And this chemical is shunted into the mitochondrial complex matrix. I was going to say something here, but now it's left me what I was going to do. And again, just keep in mind, this is an R chain, so this can be rather long. So this gets shunted in, and then what happens is that acyl group gets split back off again. And now we have that acyl CoA inside the mitochondrial matrix, where it can now be broken down further. And then we're back to just the carnitine, which can be shunted back out into the intermembrane space to be reused again. So this is just a carrier that gives us around, and it's not consumed in the process. So the ultimate goal here is to get these acyl CoAs down here into the matrix. And then what we have is what's called the oxidation spiral. This is a 4-step spiral which produces acetyl CoA, which can then enter into the Krebs cycle, or the citric acid cycle. And there's 4 particular steps in this, where it gets oxidized, then hydrated, then oxidized again, and then we can peel off this acetyl CoA. And this can happen over and over and over again repeatedly, depending on how long this R chain is. And so it's going to start clipping off 2 carbons at a time as it goes through this repeated process. And each time it goes through, you're going to get an acetyl CoA. We're going to look at this right now. So here's the acyl CoA. And we have here the carbonyl group, this carbon double bond O. And the next 2 carbons over are the focus here. This is where the cleavage is going to ultimately happen. And so this is called the alpha carbon, and this is called the beta carbon. And this also gets called beta oxidation for this reason, because we're ultimately going to oxidize this beta carbon before cleavage. So what happens is this acyl CoA gets dehydrogenated. So these two hydrogens here are going to come off, and this is done with a enzyme that's just called acyl CoA dehydrogenase. So it's dehydrogenating the acyl CoA. Remember, enzymes are just named after their function. This will take a FAD and it will pick up the two hydrogens that come off of this, and we get a double bond forming between this alpha and beta carbon. And this will always happen to form a trans isomer here. So these hydrogens are on opposite sides. And this is called trans-enoyl CoA. Any questions there? Then we take this double bond between the alpha and the beta, and we're going to hydrate it. So we're going to take this trans-enoyl CoA. We're going to use a different enzyme that's called a hydratase, and it's going to break that double bond, and it's going to put, and we have to take water, and it's going to put a hydroxy group on this beta carbon, and it's going to put a hydrogen on this alpha carbon. And the result of this is we have an L beta hydroxy acyl CoA. Then we're going to take this hydroxy acyl CoA and we're going to oxidize it again. We're going to take off the hydrogens. So we're using another dehydrogenase, and it will react with a NAD plus to give NADH, and we're going to have a free proton left over. And then what that does is that forms this double bond here with this oxygen. So we now have this C double bond O. And again, this is why it's called beta oxidation, because we're oxidizing this beta carbon here. And this is called beta keto acyl CoA. Keto because this is a ketone group, right? We've got R, C, double bond O, and then R. Any questions there? And then finally, we're going to cleave off. It's going to be done using an enzyme that's called a thiolase. And this thiolase is going to cleave the chain, leaving a acyl CoA with two fewer carbons. So we're going to lose these two carbons here, but we still have this R chain, however long that may be. And we're still going to have this carbon with a double bond O. And what happens is we now have, cleaving off this side, we have an acetyl CoA, because there's two carbons here, so it's called acetyl CoA. But then the remainder over here, this pink part, is going to react with another free CoA to form acyl CoA again. So what we've in effect done is we've pulled off two of the carbons off of this R chain, and this can now go back into step one and get processed even further, pulling off two more carbons, so forth and so on until it's out of carbons in this R group over here. So you can see here, if we had a C18 fatty acid, we can run through this cycle 8 times, and we will end up with 9 acetyl CoAs. And remember, acetyl CoAs are what can go into that Krebs cycle and produce energy. That's the whole goal of this. If you had a C14 fatty acid, you could go through this 6 times and end up with 7 acetyl CoAs in the process. So a little bit fewer because it's 2 less carbons in the chain. Any questions there before we move on? Does that make sense what we're saying here? Okay. Silence is golden. We do eat unsaturated fatty acids. The problem is that unsaturated fatty acids typically are cis isomers. And this, remember this reaction here that we saw in the first step, this forms a trans isomer. So this reaction cannot happen with a cis isomer. And so, I'm sorry, this reaction right here cannot happen with a cis isomer. It must be in a trans formation. So what your body does, amazingly enough, is it uses another enzyme. If you have, remember this is an R group stretching out here, right? If you have a MUFA or a PUFA, you've got multiple single or multiple double bonds in that R chain, then what it will do is it will bring in another enzyme that's called a cis-trans isomerase. And what it does is it actually will move this double bond between the 3rd and 4th carbon over here between the 2nd and the 3rd carbon. So it moves that double bond over here, and in the process also puts it in a transformation. Pretty amazing, huh? So it shunts it over because the reaction here is with these alpha and beta carbons. This would be the, what is it, alpha, beta, gamma, right? So this would be the gamma carbon. That's not where we want to be. We want to be on the alpha and the beta carbon. And so it shunts over and puts it in the transformation. Glucose versus tag energy production. So 3 glucoses is carbon 18, and this will give you 90 ATPs. Remember, ATPs are our energy sources, that adenosine triphosphate. Stearic acid, which is also C18, gives you about 120 ATPs. Lipids are 33% much more efficient than carbons at energy storage. And so here you see the difference. With this glucose, we get 90, but from a stearic acid, which is a lipid, we get 120 ATPs. So it's a little bit more efficient there. Our skeletal muscles use glucose when we're doing active work, and they will burn fatty acids when we are at rest. Our cardiac muscles, so our heart muscles and our blood vessels, use fatty acids first and then glucose later if they don't have fatty acids in order to consume. Our liver prefers to metabolize fatty acids. And our brains are powered solely by glucose. Fatty acids cannot cross the blood-brain barrier at all. So you can see some test questions in here like, you know, what does skeletal muscles prefer? Or what is your brain powered by? You know, or things like that. Okay, you can get an imbalance between your body when it's healthy, has a good balance between lipids and carbohydrates. You can get an imbalance in these, and this can be caused by having a diet that's high in fat, low in carbs. This is often called the keto diet or the paleo diet or the caveman diet, something like that. This imbalance can occur in diabetic conditions where the body can't process the glucose, and it can also happen with prolonged fasting. And the result of this is that you don't have enough oxaloacetate for the Krebs cycle. Remember, this is the very last species in that cycle that then continues around and around inside of there. And if you don't have enough of this, then it will take the excess acetyl CoA that our bodies have, and it builds what are called ketone bodies. And it's through a process of ketogenesis. Remember, genesis is making something. Keto is ketones. So ketogenesis is making these ketone bodies. The three main ketone bodies are acetoacetate, beta hydroxybutyrate, and acetone. You might be familiar with acetone. This is a chemical that gets used in the lab a lot to like wipe off the markers. If you marked a test tube or something, you guys ever use it for that? Or it's also used in like nail polish removers as well. Um, and I think it's used in like wasp sprays because it evaporates quickly and it actually freezes the wasp wings so that the poison stays on them longer. So the process of ketogenesis is it's going to take two free acetyl CoAs that it's not able to do anything with, and it's going to put these two together and make an acetyl CoA, and then this will react with yet another acetyl CoA to form 3-hydroxy 3-methylglutarol CoA, and then this will form acetoacetate, which acetoacetate can be further processed into this beta hydroxybutyrate or acetone. So these are our three ketone bodies that this will produce. So you're taking these excess acetyl CoAs and making these ketone bodies. If you build too many of these ketone bodies in your system, then you're going to be in trouble. So the normal level of ketones in your body is 1 milligram per 100 mils. You are in ketosis and you're in the state of ketonemia if you're up to 20 times that, or 20 milligrams per 100 mils. You are in ketosis at that point. At 70 milligrams per 100 mils, your kidneys will try to excrete these excess ketone bodies out in the urine, and this is called ketonuria. And if you are in a high level of ketosis, you can get an acetone breath because your body's just producing this acetone like crazy, and it's going to come out on your breath. Acidosis is something that is caused by getting a low pH balance in your body because you've got too many of these acetoacetates and beta hydroxybutyrates. So these both act as acids in your body and lower the pH if the concentrations get too high. And acidosis can lead to a coma and ultimately death. kill you. And this is why people do these ketone diets, is because by eating a low carb, high fat diet, and high protein, it is going to force your body to burn up your fat reserves to keep going. And so being in ketosis for a short period of time is actually good for you if you're trying to lose weight, but being in it long term can ultimately cause you to die. Any questions there at all? Okay, lipogenesis. So again, genesis is making something. Lipo is lipids, so we're going to make lipids. So sometimes if you have excess fatty acids in your body, they can reform into lipids. So this is a metabolic pathway by which fatty acids are made from excess acetyl-CoA. This happens when your dietary intake has more nutrients than you need. Lipogenesis occurs in the liver, inside of adipose tissue, those tag-containing cells, and also in mammary glands. And it involves a 4-step process where you form a ketone, then you hydrogenate it into a secondary alcohol, then you dehydrate it into an alkene, and then you hydrogenate it into an alkane. I got a picture here. I'm trying to remember what this is showing here. Fatty acid synthase. Oh, yeah. So this happens with a enzyme that's a fatty acid synthase, and there is a protein out here that attaches into this that's the carrier for this, and it will pick up these acetyl CoAs and shunt them into a long chain building a fatty acid over again. And we'll see this momentarily. Boy, I'm worried that volume's going to be really low. I don't know why it's doing that. I can just barely see the mic thing move. Oops, did I double click? Okay, so that first step is condensation, because we're going to take two species and put them together. So we're condensing them back together. This ACP is a protein that's literally called acetyl or acyl carrying protein. So here's a picture of this protein. You can see here some unstructured regions as well as some spiral regions in this protein, but it will attach to these acetyl CoAs, and it will also attach to what's called malonyl CoA, and you see these down here. Hang on, more people are coming in. And so we will have an acetyl ACP now. So you've got your acetyl group, but it's attached to a sulfur and this ACP, this protein here. And malonyl ACP is, again, you've got this acetyl group, but it's got an additional carbon with 2 oxygens on it that forms malonyl, and it is also attached to a sulfur and an ACP. And these are going to combine to where we get the acetyl portion over here attached to this part of it, and you're going to get acetyl ACP. And then you're going to have this cleaved off as carbon dioxide. And then you're going to lose one ACP with the sulfur as well, and this can be reused again back up here to react again. So we're combining these two together. Then we're going to hydrogenate this ACP. This carbonyl group is going to get hydrogenated. So we're going to get 2 hydrogens added on there. And this will form a beta hydroxybutyl ACP. So the ACP is still there, so it's still an ACP. And then we're going to dehydrate that. So we're going to pull off the OH group and form water, and we're going to get a double bond in here. I think this will be a trans double bond, and this is called crotonal ACP. And then finally, we're going to hydrogenate this double bond, and that's going to break that double bond and add hydrogens on either side to give us butyl ACP. Butyl, because we got 1, 2, 3, 4 carbons in there. And then this process can repeat over and over again. So it's kind of the opposite of the fatty acid spiral. We're going the opposite way. We're spiraling up and building fatty acids. So we start with acetyl ACP here. There's our 2 carbons. And it's going to react with a malonyl ACP. And we're going to get a C4 fatty acid out of this. So we had 2, but now we're going to have 4. Then this can react again and give us a 6. And this cycle can repeat over and over again. Here's an example where we now have a 16 carbon fatty acid. So this is lipogenesis. You're taking these excess acetyl CoAs and rebuilding fatty acids out of it. Do I have a diagram of this? Yeah. So here is your fatty acid spiral. So when you consume these fats and lipids, they're going to break down into the fatty acids. And then this fatty acids can be shunted into the mitochondrial matrix, where it goes through this fatty acid spiral to form acetyl CoAs here. But if you have too many acetyl CoAs, it can shunt them back out and through the process of lipogenesis build fatty acids again. And these fatty acids can then form triglycerols. And then if you have excess acetyl CoA also, another way it can get rid of these excess species is through ketogenesis, where it forms ketone bodies. Our bodies can produce cholesterol. Remember, we can eat cholesterol, but our bodies can also use it. Cholesterol is a C27 molecule, and it's a precursor to bile salts, the formation of sex hormones, and adrenal hormones. We talked about this a few weeks back. Cholesterol biosynthesis happens mostly in the liver, and it goes through a 27-step process that we're not going to go through in detail. You can thank me later. And it will consume 15 acetyl CoAs in order to do that. So this is eating up a lot of free energy stuff in order to build this cholesterol. But if your body needs it, then it can go through this process and synthesize cholesterol. By the way, I neglected to say this back up here, but I feel like I must. My oldest daughter had her gallbladder removed. Anybody have their gallbladder removed? You don't mind sharing? I just
I
found
have.
I just found out last night at the hospital. I didn't know this. My mom had her gallbladder removed. I either never knew that or I forgot. But this production of this bile in the, is coming from, remember, to to bring these fats into your bloodstream, right? That bile is coming from your gallbladder. If you remember, bile is stored there. And this is why, if you don't have your gallbladder anymore, you're not supposed to eat high fat diets because your body's going to have a very hard time dealing with those. All the way back down. Okay, we talked about that. And that's it for lipid metabolism. I've got some problems in here for you to look at, in addition to the practice thing we're going to do tonight. There any questions here at all about what we talked about? You guys hear that? I got background noise going on. All right, what time is it? It's almost 7. You guys want to take a short break or you want to keep going? I can go either way.
We can keep going.
All righty. Okay, so let me exit there then. Let's go over to chapter 15. Except it didn't start from the beginning. Okay. Our last piece here is protein metabolism. So, again, you eat proteins. There's no enzyme in our saliva to break down the proteins. In our stomachs, however, the proteins get denatured through the presence of hydrochloric acid in our gastric juices in our stomachs. Does anybody remember what denatured means at all? Denaturing a protein?
Well, the protein will be, if it's denatured, it's completely like changed. Like it's no longer going to be like active.
Right, it loses all of its structure. It doesn't lose its primary structure, so the sequence of the peptide bonds are the same, but it loses all of its secondary and tertiary and quaternary structure. That's right, that's the denaturing of it. So it gets all denatured. It gets hydrolyzed through a enzyme called pepsin. This is an enzyme that does this hydrolysis of the protein. And then the protein gets broken down into polypeptides. In the small intestine, we have a number of enzymes that will take and hydrolyze those peptides down to individual amino acids. And then in your intestines, you get the amino acids being transferred out into the bloodstream. So we have in our bodies what's called an amino acid pool. There are 3 sources: our diet, what we just talked about. You're eating this food and it gets broken down into these amino acids, protein turnover, and they can also be synthesized inside the liver. Protein turnover is when proteins degrade and they back to amino acids. They basically get torn apart back into amino acids. And then these amino acids can go back into to make more proteins inside the human body. Where this happens most is when you're using your muscles, you're going to wear down your proteins that make up those muscles, and some of them will go revert back to amino acids. In a healthy adult, about 2% of your proteins turn over every day. So 2% of your proteins revert back to amino acids and go back into the amino acid pool. Disease can also cause this, or injury, or, like I said, normal wear and tear on the body. So this is the process by which the body can restore itself. And this is what's happening, like if you're a bodybuilder, right? You're breaking down your muscles and they will rebuild, but they will always rebuild stronger and therefore larger, because you're stressing out those muscles in order to do that. So we have these amino acids in our body. What do they get used for? About 75% of them get used to make more proteins. So, that's why we eat protein, is so that we can break it down to amino acids and make more protein. But they can also be used to make non-protein nitrogen compounds, such as purines and pyrimidines. You remember these figure heavily in the formation of DNA and RNA. Hemes, which is the active part of the hemoglobin that carries oxygen through our body. They are also neurotransmitters. And choline is made up of these nitrogen compounds, as well as epinephrine, or adrenaline, also comes from this. So we can use this available amino acids in our body to form these nitrogen containing compounds that are not proteins. We could also use them to form non-essential amino acids. Remember, essential amino acids are the ones that you have to eat. Your body can't produce them, but our bodies do make non-essential amino acids. And so these free amino acids can be used to build these things up. And also used for energy. So these amino acids can be consumed, where the nitrogen gets removed and the remaining carbon skeleton gets converted to pyruvate, acetyl CoA, or some other citric acid intermediate. And we'll look at this momentarily. So here we have the amino acid, and it gets broken into a nitrogen portion and a carbon portion. This nitrogen portion can be eliminated from the body via urea. It can also be used in the biosynthesis of non-essential amino acids, like we just said, and also in the biosynthesis of these nitrogen-containing compounds that are not proteins. The carbon portion can get broken down, and it can be converted into triglycerides, and by creating fatty acids. It can be broken down and formed into glucose through gluconeogenesis. Remember, we studied that a while back. They can be consumed by the citric acid cycle and produce ATPs. these energy containing units, or they can also be used to form ketone bodies through ketogenesis. Any questions there at all? So, amino group release. So, to get rid of, to cleave off this nitrogen portion, it goes through a transamination process, which moves the amino group to another molecule. It then goes through a deamination process where the amino group gets removed from that other molecule. And that results in ammonia, which is converted to ammonium. And the reason why is ammonia is very toxic, so your body is going to convert it to ammonium right away. And then it will convert the ammonium to urea via the urea cycle that we're going to look at momentarily. And then your body will excrete this urea out in the urine. So transamination, like I said, is moving this amino group to another amino acid. And how this process happens is we have an alpha amino acid with this amino group on it. It will react with a keto acid. Remember, you got this carbon double bond O that makes this a keto acid with the COO minus over here. And you've got these two R chains on the sides. And what's going to happen is that this will go over onto this carbonyl group here, forming a new alpha amino acid and a new keto acid. So they just kind of switch places, if you will. This involves a transaminase enzyme. There are about 50 different kinds in our bodies. Most of these reactions use a species called alpha ketoglutarate. And this is alpha ketoglutarate right here. And so here in this example, we have alanine reacts with this alpha ketoglutarate with the enzyme catalyzing the reaction. And you end up with pyruvate, which remember is part of the, can be fed into the Krebs cycle, and glutamate, in this particular example here with alanine. Then what happens is that we're going to release that, let's see here. Oh, I'm sorry, this is back. Another part of this, how this process happens with this transamination is this transaminase has a functional group on it. So remember, these enzymes have large proteins, but the active site is this species called pyridoxal phosphate. And here you see it right here. So in our example, we have alanine. It reacts with this active site of the enzyme, and we end up with pyruvate, and then we end up with the amino acid going onto this carbonyl group right here. And then this reacts with that alpha ketoglutarate and forms our glutamate, and we end up with back to the pyridoxal phosphate. So this enzyme ends up back in the same state again, and we have moved this ammonia group from one species over to another. Then this new species we're going to deaminate, not deanimate, that'd be killing it. That was a joke. It's deaminate. So what happens here, in our example, we have glutamate. It reacts with a NAD plus and water. And again, another enzyme is involved. And we end up with this free ammonium that we talked about. And we're back to our alpha ketoglutarate. And we now have NADH and a free proton. So the net reaction of this, and this alpha ketoglutarate can be reused in the transamination cycle again. The net reaction is we take an alpha amino acid, we react it with NAD plus and water, and we get an alpha keto acid, free ammonium, NADH plus, and a free proton. And then, like we said, this ammonium is going to get eliminated via the urea cycle, which we're probably talking about in a second. Both the NADH plus and H plus can be used in that electron transport chain to produce more ATPs. And yes, we're at the urea cycle. Okay, so this is a high-level overview of the urea cycle. We've got the green area out here in the outer part of the cell. This is the cytosol. And we have inside of the mitochondrial matrix, just a couple of the steps in the process happens. The rest of them happen out into the cytosol. And what we're going to do is react our species with, go through a transfer process to form something called citrulline, which can be shunted out into the cytosol and continue through this process, which ultimately ends up with urea that gets excreted off. And then we're back to ornithine, which can shunt back into the matrix to be reused over again. So this ornithine gets changed over to citrulline, gets shunted out into the outer part of the cell, gets processed around, and can come back in again. So this is why it's called the urea cycle. So the first step is we have this free ammonium inside of here, and it will react with carbon dioxide, water, and it's going to take 2 ATPs. So this is consuming energy, and it forms this carbonyl phosphate. So we get a phosphate off of this ATP. That's this group right here. And remember, the tilde denotes a strained bond because of these highly electronegative oxygens here. We now have this ammonium attached as an amino group onto this. And we're going to have our ATPs drop to ADPs. We're going to have 1 free inorganic phosphate and 3 hydrogen ions as products as well. Ornithine then, so ornithine right here is going to come across and react with this carbonyl phosphate that we just made. And it's going to attach, it's going to break off that phosphate side, that strain bond. So we're going to get free inorganic phosphate. And this portion is going to get attached to the ornithine on the nitrogen. And this gives us citrulline. This can be shunted via a protein out of the mitochondrial matrix and out into the outer part of the cell. Then this citrulline is going to go through a condensation reaction. So that means we're going to take these two and add them together and form one new molecule. And it reacts with aspartate. So here's aspartate. Again, this is going to consume energy to put these together. So we have two inorganic phosphates coming off from a triphosphate down to a monophosphate. And we're going to have this guy plug right in to the oxygen up here. And this is going to give us arginine succinate. Again, I'm not going to test you guys on these kind of details, but understanding this overall process is helpful. The succinate then will get cleaved off from there. We will cleave off this portion here, and this is fumarate. Fumarate, if you recall, is one of the intermediates in the Krebs cycle. So this can feed into the Krebs cycle, and we're left with arginine here. This arginine then gets hydrolyzed with water, and we form urea. So urea has got urea is one carbon with a double bond oxygen and two nitrogen groups, two amino groups here. This is urea. And then we're back to just ornithine, which can then get shunted back into the mitochondrial matrix. This urea then can get expelled off through urine and be gone from the body. So that's how we're getting rid of this toxic ammonia product. That's the whole goal of the urea cycle. The aspartate in that first step comes from the Krebs cycle. Fumarate, like I said, is an intermediate product in the Krebs cycle, so it can be consumed there. The net reaction of the urea cycle overall is we've got these nasty ammoniums that we're wanting to get rid of. Two of those will react with carbon dioxide, 3 ATPs, 2 waters, and aspartate, and it will give us urea, 2 ADPs, an AMP, and 4 free inorganic phosphates and fumarate. Any questions there at all? So here you can kind of see it in a conceptual diagram. Here we have this citric acid cycle, and one of the intermediates in the citric acid cycle is this fumarate. So when we produce fumarate over here in the urea cycle, it can be fed into that citric acid cycle to contribute to that whole process. But oxaloacetate is an intermediate in this citric acid process, and it can react with glutamate to produce alpha ketoglutarate and give us aspartate. And this aspartate can be shunted into the urea cycle to be that reactant that we need in order to keep this cycle going. And then, of course, this cycle processes through and gives us the urea off that we don't want. We want to get that out of our bodies. And so you see these two cycles feed each other. That might be a good test question too. You know, like what are the two species that connect these two cycles together or something like that. Have you guys studied this before, like in biology classes or something? Or is this all brand new?
I don't think I've studied this cycle.
You have or have not?
Have not.
Oh, okay. I was just curious. Okay. But remember, we have that carbon portion of it. So we've now dealt with the nitrogen portion of it. We now have this carbon portion that comes from the protein. There are many different pathways to degrade that carbon skeleton. If we have glucogenic amino acids, they will produce glucose. If we have ketogenic amino acids, they will produce ketone bodies. And again, the hint is in the name. Genesis means to make, right? So glucogenic is going to create glucose. Ketogenic is going to produce ketones. And so what we have here are, and again, we're not going to go into all the details of all this, but you can see here that the ones that are colored green are ketogenic. So we have isoleucine, leucine, and tryptophan are all ketogenic, and they can be broken down to give us acetyl-CoA, which can be fed into the citric acid cycle, but they can also be used to create ketone bodies. These species are also ketogenic, and they will form acetyl-CoA, and it can be then converted into acetyl-CoA and then go into either the citric acid cycle or form ketone bodies. The blue ones are glucogenic. These, I'm drawing a blank here. Yeah, sorry. These amino acids are going to form glucose-related compounds, right? So here we have like glutamate, glutamine, histidine, proline, and arginine will all form alpha-ketoglutarate, which is an intermediate step in the citric acid cycle, which can then come through that cycle, form oxaloacetate, and that can be excess oxaloacetate. We studied this a few weeks ago, can go and form glucose through glucogenesis. We'll see an overall diagram that shows all this as well. The same with these guys. These guys can feed in, they can be, the pathways for these will produce oxaloacetate. These guys over here can form fumarate, which then gets inserted into the citric acid cycle, and all those can ultimately feed back out and form glucose. There are a few amino acids that can do both. And so they can be reduced to pyruvate, and then pyruvate can either go into the citric acid cycle, or it can go to form acetyl-CoA to go into the citric acid cycle, or it can go and generate ketone bodies. So these guys can go either way. So the ones with asterisks on them are the ones that have multiple pathways into those processes. Any questions there at all? Okay, we can also build amino acids in our bodies. There are 11 non-essential amino acids, and our bodies can build these. And glycolysis has 2 intermediates, 3-phosphoglycerate and pyruvate. Pyruvate can be converted into alanine, and 3-phosphoglycerate can be converted into serine, which can then be converted into cysteine and glycine. So these four amino acids can be created from byproducts or intermediate products of the glycolysis cycle. Remember, glycolysis is breaking the glucose down. Two intermediates of the citric acid cycle are oxaloacetate, which we were just looking at, and alpha ketoglutarate. And these can be used to form all of these amino acids. So it kind of reverses that process we were just looking at. And then finally, there is an essential amino acid that we have to eat. That's phenylalanine. And this can go through a pathway to form tyrosine. So this non-essential amino acid can be created from this essential amino acid that we have to take into our bodies. Hemoglobin catabolism. I always feel like I'm saying that wrong. Catabolism, catalysis, catabolism, catabolism. I think it's catabolism. Does that sound right? Okay. Our red blood cells have hemoglobin in them. Red blood cells are formed in bone marrow. Our bodies produce about 200 billion red blood cells a day. Their lifespan is about 4 months. And they do contain hemoglobin. Remember, hemoglobin carries oxygen to throughout the body, and it also removes carbon dioxide. And it is made up of these 4 pyroles that have an iron setting in the middle, and it's this iron is the active site that carries that oxygen throughout the body. Okay. So old hemoglobin primarily gets broken down or degraded inside the spleen, but it can also happen inside the liver. The protein portion, so remember, this is just the heme. There's a whole protein around this. The protein portion can get converted back to amino acids and enter into the amino acid pool. The iron that's freed up when this breaks down gets stored as ferritin, and it can then be used to form new hemoglobin. So ferritin is this huge globular protein that holds the iron inside of there. So here's these iron atoms. They get trapped into what are called nano cages inside of these large globular proteins, and it's held there until it's needed to form new hemoglobin. The tetrapyrrole structure, so that's this 4 pyroles here, get converted into bile pigments, and these bile pigments can be eliminated as either feces or urine. Jaundice is a condition where there's an imbalance with the bilirubin levels are very high in our bodies, and this causes yellow eyes and yellow skin. And bilirubin is formed in the degradation of hemoglobin. We're going to look at this in a moment. So as this old hemoglobin gets broken down, we form this intermediate species bilirubin, and if those levels are too high, it causes what's called jaundice. So the first thing that happens in the degradation process is that we break a bond and remove a carbon. So this heme in this nice little ring, multi-ring structure here, this gets pulled out, and notice that you now have, it reacts with two oxygens, and you end up with a double bond oxygen, but these oxygens are not connected to each other. They're not bonded to each other. So this ring is now not completely closed. And this is called biliverdin. This biliverdin, just drawn here in a chain format. So here's that double bond oxygen that we saw right here. We just sort of took this and stretched it out so you can see it a little bit better. What happens is that we take this pyrrole backbone here, and where it has a carbon double bond with one of these pyrroles, and we break that double bond and we form single bonds with this carbon. So you're going to have a hydrogen and a hydrogen here. And this is bilirubin. And for shorthand's sake, we just talk about, we just put letters on here. Methyl, of course, is a CH3 group. Vinyl is a C double bond C group. And propionate is 3 carbons, and you've got these 2 oxygens with a negative charge to get the nate part of that name. And so here we have these across here. They're not really involved in this reaction at all. It's just the breaking of this double bond right here, and the biliverdin turns into bilirubin. This is the species that if it builds up in your body can cause jaundice. When I was a kid, we actually would get bilirubin tests. I don't think they do that so much anymore. Probably you guys didn't get any of that. But I never knew what bilirubin was, but we always heard about bilirubin tests. This bilirubin gets moved to the liver, where it combines with some sugar residues and then gets transported to the small intestine. So we've got a couple of sugars here, and the bilirubin forms a bridge between these 2 sugars, forming bilirubin diglucuronide. This gets shunted to the small intestine, where, and I think this is really interesting, bacteria in your small intestine will convert it into either urobilin or stercobilin. And urobilin, as you might guess, gets excreted from the body as urine, and stercobilin gets excreted as feces. So we have to have this foreign bacteria in our small intestine in order to make this happen. And now that I think about it, that's probably the bacteria that's missing in infants. They used to give infants bilirubin shots. I think I brought this up in my last class. Has anybody here worked as a nurse or like a prenatal nurse or anything like that? I think in my last class somebody was. And I can't remember what she said, if they still give these shots to babies or not.
I know they put them under the UV lights if they are low in bilirubin.
Oh, do they really? That's if they're low or high.
Low. Or wait, no, I'm sorry. That's if they're high. If they're high. Because they're jaundiced.
Yeah. Okay. Yeah. So I wonder if it's newborns sometimes are lacking this bacteria, and so the bilirubin's building up. I'll have to look into that. I don't remember if anybody told me in previous classes or not. Okay, so here we see the whole shebang. I think we're at the close to the end here. So this shows you the lipid, carbohydrate, and protein metabolism all in one beautiful diagram. So we consume lipids. These can break down into fatty acids. These fatty acids can go through that fatty acid spiral to form tons of acetyl CoAs. These acetyl CoAs can then be fed into the citric acid cycle, which ultimately produces ATPs for energy in the body. If we end up with excess acetyl CoA, we can go through lipogenesis to rebuild fatty acids in order to get rid of these excess acetyl CoA. We can also form glycerol from these dietary lipids. This glycerol can go through glycolysis and form pyruvate. Pyruvate can be transferred into acetyl CoA, which can feed that Krebs cycle as well. Our carbohydrates basically break down into glucose. That glucose can then be broken down through glycolysis into pyruvate, therefore forming acetyl CoA and going into the citric acid cycle. Excess pyruvate can go through gluconeogenesis. Remember, neo is new, and genesis is making something, so it's making new glucoses. If we have too much of this, it can go back into glucose. Our proteins get broken down into amino acids, and then they can be broken down further, and we get the ammonia portion here, which goes through the urea cycle to form urea, and the urea gets expelled off. It can also form fumarate that can be fed into the Krebs cycle. And the Krebs cycle produces oxaloacetate, which can be formed into aspartate and feed into the urea cycle. So that's that connection between those two. These amino acids that got deaminated, so it doesn't have the ammonia anymore, forms these alpha keto acids. And these can go directly into the Krebs cycle. They can be converted into acetyl CoA, where it can go into the Krebs cycle. They can also be formed into acetyl CoA. So this is 4 carbons. And that can be broken down into acetyl CoA. They can also be processed into pyruvate, which can then also go in this direction or back up in that direction. So we see how the whole thing kind of gets put together, which is kind of cool. You sort of see it all there. Any questions there at all? So I thought this slide was kind of interesting. If you're feasting or overeating, as I'm wont to do, your ingestion of food exceeds the energy needs in your body, and this is going to cause your body to store some of it as glycogen. Remember, those are those big multi-chain polymers of glucose, and the rest of it is fat. And if you're eating carbs, you're going to get glucose, and that can be stored as glycogen in your liver and muscles, but it can also be stored as body fat. Fats that you eat can produce fatty acids, which can also be stored as fat. Proteins that you eat break down into amino acids, but they can produce fat as well, or you can lose out that nitrogen portion through urea. If you're fasting, like people do when they're doing the intermittent fasting, you have no food coming in, then your body is going to use up stored fat and glycogen in order to get energy. So the glycogen, which is stored in the liver and muscles, will get broken out into glucose, which can feed your brain, and it can also feed overall body energy. So this glucose then kind of goes back into this whole process and gives us energy. Body fat can be broken down into fatty acids, and then that can be processed into energy. So we get our fatty acids getting put into here and giving us ATPs. If you're starving, if your fasting goes beyond your glycogen reserves, then remember we said glycogen reserves lasted about 30 to 40 days. When they're depleted, then your body is going to start eating proteins to get energy. So this is why you lose body mass. It's not just fat anymore. You will consume any fat left into fatty acids to ketone bodies to produce energy. But it's going to start eating your protein, which is not good, to make amino acids. These amino acids can produce ketone bodies. It can be the nitrogen portion of it can get expelled as urea, but you can also form glucoses to power the brain and also for overall body, energy in your body. So it starts basically eating itself to keep your systems running. And if you go too long like that, of course, you're going to die because it will run out of fat and proteins to process, and then your body's not going to have any energy to do anything. Any questions there at all? And we have here, again, some more problems for you as well to look at. You'll see from these problems, again, I'm not asking you to memorize all these complex reactions or to be able to draw structures or anything like that. I'm not going to be asking you to do that. I'm going to pause sharing here for a minute. And why is it asking me that?
Will our practice test run through Tuesday?
Yeah, after we go through it tonight, I will fire it up again for you guys like I did the last ones, so you can run through them again. And,
Thank you.
and I realized today, I'm not sure if I have chapter 13 questions in there or not. So I may have to add some too. Oh, somebody's coming in. All right. Here, I'm just going to stop.